DNA replication is a super important process by which DNA makes an identical replica of itself during cell division. This takes place in the nucleus. This process is made possible by a few key players. Nucleotides. This is the basic building block of DNA. Nucleotides are made out of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, while cytosine and thymine are pyrimidines. It is also made of a phosphate group and a deoxyribose sugar. These two together make up DNA's backbone. More key players are enzymes. There is primase, the initializing enzyme, helicase, the unzipping enzyme, DNA polymerase, the builder enzyme, ligase, the gluing enzyme, topoisomerase, and single-stranded binding proteins. This is a double-stranded DNA about to go through replication. Double-stranded DNAs are anti-parallel. The upper strand, which is the leading strand, runs 5' to 3', while the bottom strand, the lagging strand, runs 3' to 5'. The strands are complementary to each other. A always binds with T and G always binds with C. This is made possible by hydrogen bonds. The first step to replication is the unzipping of DNA. The helicase enzyme does this by breaking through the hydrogen bonds. Single-stranded binding proteins then bind to the bases to prevent them from bonding back together. On the part that has not been unzipped, topoisomerase binds to prevent supercoiling. The enzyme primase then initiates the replication by producing the primer, the starting point of the replication. These are RNA bases. Since the leading strand runs from 5' to 3', its replica will run from 3' to 5', which is the only way DNA polymerase can build from. DNA polymerase also proofreads the bases as it builds to prevent any mistakes. The leading strand will replicate continuously. Meanwhile, the lagging strand cannot build continuously as it runs from 5' to 3'. It will need to have a primer every time it builds a fragment. These are RNA bases. This then creates Okazaki fragments. The enzyme exonuclease then removes all RNA bases, and DNA polymerase replaces them with DNA bases. DNA ligase then goes through both the strands to bind any gaps, especially ones made in the lagging strand. We are then left with 2 identical double-stranded DNA that are semi-conservative, meaning one strand is old and one strand is new.